we're really excited about the Regenerate Now program that we've been piloting while we've all been in lockdown. It's part of a bigger program that we have been generating together as a community over the last year. We have an Urban Farmers Alliance WhatsApp channel that is giving daily mentoring, peer-to-peer mentoring via a WhatsApp channel that gives people an opportunity to ask a question while they're in the field. We also have a Regenerate Now Instagram where our early startup farms are actually teaching people while farming in the field. And then we have a Farmers in the Field mentoring session on a 4 o'clock on a Friday afternoon in New Zealand, where those farmers get a one-on-one mentoring session with our Regenerate Now farmers, mentors. So to pull this all together, we've created this Masterclass series, which gives us a chance to enable you to do some really in-depth learning around how the biological engine works. The first masterclass that we did a couple of weeks ago really looked at soil systems. And today I'm really excited to be able to share with you this wonderful adventure of the microbiology in the soil systems that makes everything work. So today Daniel is going to take us on a journey particularly looking at fungal networks. You can use the chat channel as we're going through the masterclass to ask questions. And for those of you on the UFA WhatsApp channel, you can continue to have a conversation and learning with us after this particular session. This will be recorded and archived so that anyone can actually watch it. To you, Daniel.
Good evening and welcome to all those people that have joined us. Hopefully you'll enjoy this little quick tour through the soil microbial system. We're focusing primarily on fungi tonight as you watch through it. So I'll try and see if I can get this thing to work to share the screen and we'll get started. Oh, hopefully we're all good. So there's a continuation from last week's healthy soil systems. Let me get everything working. Right, here we go. And the principles that we're working with are what we call biology first principles. And we say the healthy soil systems are all about balance and do no harm biology first principles. Last week, the first masterclass, we did a quick overview of how the soil system is designed to function. So explaining what is soil and nutrient cycles and organic matter, soil structure, soil erosion, and examples of healthy soils. It actually says spoils, interestingly enough. Somewhere I must have left a typo in there. If you haven't actually seen it, it'll help you actually join together some of the dots that we're probably going to create for you tonight. This second class is focused on the Rhizosphere microbial system, which basically means everything, or the area around the root zone of a plant is called the rhizosphere. The area around the leaf of the plant is called the phyllosphere, and that's what we'll be talking about next week, or the next masterclass, I should say. We're going to go through a very brief history of damage to the rhizosphere, and we're going to talk about the key fungal organism, mycorrhizal fungi in our soil systems. And then we look at things like fungal to bacterial ratios and their importance, what really drives plant productivity, so getting people understanding how to drive their soils to be more productive, and lastly, the importance that fungi plays in the carbon cycle. So I've got about 20 slides to go through, and so feel free to ask questions as we go along, but I'm going to try and get through them on time this week. So I've put this slide back in again because it's so important to understand the things that we do that are negative to soil health. Things like monocultures, pesticides, herbicides, insecticides, fungicides, including the organic ones. As I said last week, it's not discriminatory. If it as a broad spectrum side, it basically means to kill, it's death. So it's obviously not going to have a positive response for our soil system. The same with sterilisation and solarisation. Chemical or heat treatments are all negative to this soil system. Particularly cultivation or tilling, as many people know that these days. So ploughing, rotary hoeing, power harrows, ripping. It was an interesting thing I saw today is actually the difference between running a plough behind 2 oxen versus behind a 400 horsepower tractor. There's no question that the tractor does more damage. So, you know, although we've been ploughing for a very long time, we've actually managed to make it worse. Fallowing, which is leaving soil bare for extended periods, a very common practice in broadacre and vegetable production, where a paddock is left with nothing growing in it. Usually it's herbicided off, often for months and months on end, while they wait for the conditions to plant the crop. They're doing that at the moment with onions and pukakoas. While you see all those bare fields, they rotary hoed them up and got them ready months ahead of when they could plant them, which they'll be doing shortly. Excess fertiliser and inputs, and that includes any inputs in excess, including compost. So all these practices have one thing in common, is they harm the microbial life that's the basis of the soil system. And these are very common in both conventional and organic farming systems. Now, this is giving some indication of the degree of damage that we're actually doing on an annual basis to our topsoil around the world. Unfortunately, I couldn't correlate exactly the same unit of standard of measurement that they were using in this graph to be able to recreate it and put New Zealand alongside it. But it gives you some idea of, obviously, what we're looking at here is new soil is being produced, but on a global scale, it's very small in comparison to the amount of soil loss. We're basically talking, obviously, topsoil loss here. If, for example, you're looking at US cropland, US rangeland, basically grasslands, pastoral type farming systems, and then a comparative with India and China, you look at the amount of loss. So we're looking at something like, they estimate it's 75 billion tonnes of fertile soil lost every year, which is where you'll see later on, we talk about the prediction is that we only have 60, 65 years left of topsoil if we keep losing at the rate we do. New Zealand, in many respects, is not that different. We just have a lot smaller landmass to lose soil from, but our 192 million tonnes of soil per year is lost, and that's according to MFE and Statistics New Zealand published in 2018. So it is, as I explained last week, there's only one reason that the soil can wash away and blow away. It's because it's no longer soil, it's become dirt, and there's nothing holding it together. So a brief run through the soil food web again. I found a slightly different graphic from last week. Hopefully it tries to explain it a little bit better. There's lots of these. There's always fine gaps in them that I think they should have connected. But basically what we did last week was discuss essentially what's happening in this zone with the plants and the parent material and the organic matter and this basic operation of how the other organisms were critical to making that soil system work. And today we're really only going to talk about the fungi and a little bit about the relationship with bacteria and some of these other organisms. But in 30 minutes, I just decided there was no way I was going to have an in-depth conversation about nematodes, beneficial protozoa, and all the other variation of organisms that actually live in the rhizosphere. We'll just have to put those on for another discussion. Essentially, what we're looking at is bacteria and fungi form a massive wall around the roots because the plants feed them. So protozoa and nematodes are attracted to that large number of their prey. They're feeding on the bacteria and fungi, and because the nutrients are so much higher in bacteria and fungi than the predators consuming them, those excess nutrients are released, but in plant available forms. So the soil food web came up with a terminology called the poop loop, which is a way of describing essentially this nutrient cycling that happens. So it's looking here at getting people to understand, so you have living plants, they die and you get this dead organic matter, and it gets, so as a plant gets then decomposed by saprophytic fungi, aerobic bacteria, there's nitrogen accumulation happening there, so the plants are being broken down, and the bacteria and fungi need that nitrogen to multiply, so you get the nitrogen carbon being, at this point, is now locked in the microbial system. You get microscopic soil predators like protozoa, amoeba, flagellates. There's 3 different sorts of protozoa, amoeba, flagellates, and ciliates. They feed on those organisms, and they basically poop out the excess, which in this case is the nitrogen, because they're, as I discussed last week briefly, I think that their carbon nitrogen ratio is like 30:1 versus sort of 5:1, 10:1 over in their food sources. But then those organisms get eaten by predators, and those predators, so they're eating the protozoa, like earthworms, but then they poop out the excess nitrogen, so you get more nitrogen. And this when they basically have excreted these exudates, this poop loop, it's that poop that the living plants can access. But while that mineral nitrogen is actually in the organisms, it is trapped. It can't wash away, it can't flood away, it can't wash into waterways, it can't pollute the system. That nitrogen at this point, when it's in the bodies of these organisms, is physically trapped. Even if it goes into waterways, it will not pollute with nitrates. It's another aspect to the cycle that we'll discuss later on. And so it just basically goes on. One predator eats another predator until you get to vertebrates like burbs, eating worms, and it all combines to the poop loop. So hopefully we've got some better idea on nutrient cycle. Going in depth into the soil, we start now looking at the mycorrhizal fungal aspect of it. And there's 2 types of mycorrhizae. One's called an ectomycorrhizae, which is what we're showing here, mycorrhizal fungi. I'll show you what the differences are between them. This is showing an example of a pine seedling, and all these fine filaments out here are actually not roots. These are showing the mycorrhizal hyphae, the hyphae from the fungi stretching out. So you can see there's calculations at times with the plant roots, essentially are these structures that we're looking at here, with the fine root tips on the end. They can feed here. There are calculations out there that they say the mycorrhizal fungi can increase the root area, effective area of the plant, to be able to obtain nutrition by up to 10,000 times what the original root capacity of that plant was. So that's where we talk about how much more the soil can get nutrients from when you have mycorrhizal fungi working as it's supposed to on most plants. We'll talk about some of the plants that don't have mycorrhizal fungi, but with the ones that do, that makes an enormous difference. In the early days of the Pinus radiata plantations in New Zealand, when they first brought them here, they couldn't get the pines to thrive, apparently, and it turns out very early on, a very long time ago, they worked out that they actually had to inoculate the pines with mycorrhizal fungi to actually get them to grow at all in New Zealand conditions. If you just planted them out there, there was no. They're very, in this case, a quite specific group of mycorrhizae that work with them. Ectomycorrhizal fungi only work with about 10% from memory of plants. Most of the plants, particularly most of the food-producing plants, apart from brassicas, use endomycorrhizal fungi. And where the ectomycorrhizal fungi form a layer outside, with another graphic, I'll show you that. The endomycorrhizae, you can actually see, so what we're looking at here is a root system. The brightly coloured colours on the inside are actually the fungi inside the root. So this is what's known as a vascular arbuscular mycorrhizal fungi. It's a particular group of mycorrhizae. They belong to the endomycorrhizal family, so growing inside the plant cells. So a lot of they actually invade the actual cell itself, and this is how they form that symbiotic relationship with the plant. This graphic is showing us the difference between endomycorrhizae and ectomycorrhizae that I just talked about, and it very clearly shows with ectomycorrhizae, you have a lot of the fungal hyphae on the outside of the root, and the ones that come into the root actually go in what we call the intercellular spaces. So that in between the cells, there are gaps, and they get in between those spaces. But the endomycorrhizae, actually you can see they're actually in the cell. They're getting into the cell of the plant itself. And it's just the difference between the relationship. So a plant that is meant to form a relationship with an endomycorrhizal organism will not form a relationship with an ectomycorrhizal organism. Although I'm sure there are cases where both of these exist on the same plant, but as a general rule, we're looking at one or the other. So if you're looking at inoculating your vegetable production, for argument's sake, the plants other than your brassicas, what you're looking for is that the mycorrhizae we should be using is endomycorrhizae, which is the AMF vascular mycorrhizal, or the VAM mycorrhizal endomycorrhizae, which is what you find in the Nutrilife Network product that many of you actually have been using. There are ectomycorrhizae product on the market, but it would be a waste of time if you were using the wrong mycorrhizae for a particular species. Brassicas.
So can I ask you a quick question, Daniel? So what you're saying is that certain plants will form a relationship with one or other, and how many, I mean, in terms of, I'm sure you don't know how many different types of ecto and endomycorrhizae there are, but is there a kind of a range, just to give a sense of the you know, the vastness of this type of fungal group?
I had a guess. I think I've seen kind of about 100 different species, but to be fair, there's no way that we have any idea in the science world how many exist, because the identification of these is happening all the time, and that number would be changing. I mean, to give you an idea of how little sometimes we do know, is something as huge as an organism like a nematode, which is something you can actually see sometimes with the naked eye. These are all tiny microscopic organisms. Something as large as a nematode, they believe they've identified 20,000 different species of nematode, but through DNA analysis, they know there's about another 80,000 that they know exist, but they actually don't know what they look like or how to actually identify them beyond the DNA fingerprint that they find. And that's a very common finding they have these days within the microbial world. They use DNA analysis to find out who is present. They can't tell how many are present with that analysis system, but they can tell who is present. The problem is that quite often, even in the most common places, they find 50, 60, 70% of the DNA comes back from organisms that they have no ability to identify. There's been all sorts of interesting studies with that, taking scrapings off subway walls in New York and doing DNA analysis and finding about 50% of the organisms present on the wall of a subway. We have no idea what they are or what they do.
Can I ask you another question?
Sure.
Because I've known about mycorrhizal fungi for quite some time, but I really have known more about the ecto rather than the endo. What I understand about them is that they they sort of operate like a bit like a hair extension on the plant. And is that the same for the endomycorrhizae as well? Because I'm noticing that even though it establishes itself differently within the cell structure of the plant, does it still actually have a part of it that is in fact, you know, moving into the soil system?
Yeah. So what what we're seeing in the diagram here is these hyphae that are outside the plant wall. Okay, so this is essentially a side view of a root, and so you're showing hyphae going into the root and into the cells, but what's actually feeding the mycorrhizae that are there, the food that they're finding that to actually feed the plant with, in this case, they're actually getting from the soil system outside the root. And whether it's endo or ecto, we're still talking about this thousands of times its root mass is actually being presented, is actually being created with all these fungal hyphae. It's these sorts of fungal hyphae also that can connect individual plants together, so you can get, and what they talk about is in a forest system, a large plant's mycorrhizal fungi, they'll actually virtually farm the younger plants in and around and under the trees that maybe don't have the light resources that the higher canopy trees have. So they actually feed those organisms as well in, I guess, this mutualistic system, so you can get healthy trees feeding sick trees through, and it all happens via mycorrhizal fungi, big trees feeding small trees. This is really the fungal network that we're talking about, and it stretches literally from one side of of the field to the other in between the plants. And you can imagine all these fungal hyphae in the soil, how much damage we do when we start running things like cultivators or plows through, because these fungi hyphal networks are hugely extensive. Does that answer the question?
Great.
Okay. Right. So now we're going to move on a little bit further, looking at the relationship between, and this is where we start talking about a fungal to bacterial ratio. So there's what I want to show you is the importance of balance in the soil system, because obviously this soil food network needs all these organisms operating kind of in sync, in balance. And if you have them, if one group of organisms isn't present, then you really struggle to make the other organisms function. So we need to find a balance. And what we're looking at is a consequence of when that balance is out, of what sort of things happen, what are the positives and negatives associated with this. So when we're starting with the bare parent material in the beginning, the start of that process is basically 100% bacterial. There isn't any fungi at that point. As the system starts to evolve, we start having things like cyanobacteria, which we won't go in today to explain what those are. Just haven't got the time. True bacteria, protozoa, fungi, nematodes, and microarthropods. But in this situation, we've got a fungi to bacterial ratio, which is very, very heavy still in bacteria and very low in fungi. When we've got this sort of level, so we've improved it a little bit, but it's still massively in favor of bacteria. What we find is we have very high nitrate levels, and I'll explain later exactly why that happens. But essentially, it's a function of that microbial system that we end up with these high nitrates. That sponsors weed growth. And I'm pretty sure we've got, we can later on, we'll talk about, or in some of the other masterclasses, we'll start talking about actually how our fungal to bacterial ratios can actually help us manage weeds. But when we've got very low fungi and high bacteria, we tend to have much higher weed issues. Then we start getting through to what kind of needs to support it. So as I said, brassicas are not requiring things like mycorrhizal fungi in this case. So in a low fungal to bacterial ratio, so we're still only at 0.3, so we're talking about, there's basically 3 bacteria, 3 or 4 bacteria for every fungi. Early grasses, brassicas, mustards, that's the sort of situation that's happy, those plants happy growing. When we start improving the fungal level, we start being able to support different sorts of grasses and vegetables. It becomes much more productive in that. And then when we start looking at closer to this sort of almost 1-to-1, sort of 0.75 to 1-to-1, we're starting to look at really healthy sort of row cropping, pasture lands, soil environment. But if we're wanting to grow more substantial plants, things like shrubs, vines, and bushes, then we're starting to look, and we actually need to go the other way. We need to go more fungal. So the more evolved, the slower growing, the longer those organisms are there, the more reliant they become on fungi. So you can see here, we're already at 5-to-1. We go to deciduous trees, we're leaping up to 100-to-1. And then we start getting into what they call old growth conifer forests in this case, because it's out of the US. and we're looking at massive fungal dominance, and this is how that forest system gets supported. So when you start having all these different level plants growing, and the necessity of that fungal network to actually support the plants that are lower down, younger in the system with a massive high canopy trees, basically the older it gets and the taller those trees get, the more you actually need fungal to move that food source around the forest floor.
So just to have clarification for one of our viewers, the ratio of, like, you've got shrubs, vines and bushes there, you've got a ratio of 2 to 1 and 5 to 1. What you're talking about is a ratio of fungi to bacteria. So it's not actually a ratio of nutrients, it's actually a ratio of fungi to bacteria. So each of those, you've started with very low ratio of fungi to bacteria, and as we've gone around the cycle, the ratio of fungi to bacteria has increased, and so has the capacity of that soil system to support different types of plants. Is that correct?
That's correct.
Okay, great.
So what we're looking at here is an understanding of how that microbial system works in terms of soil conditions and what organisms thrive in different soil conditions. The top figures up here, the 4 ppm and 6 ppm, are talking about parts per million of oxygen, essentially, in the soil system. And what we're talking about here is anaerobic conditions, so this really lacks oxygen. The facultative group of organisms in the middle here, they can live in either the anaerobic condition or the aerobic condition. The aerobic condition is where most of our beneficials actually thrive. An interesting thing from here is most of our pathogens or disease-forming organisms, and it's pretty much the same with a human body, thrive in what we call anaerobic conditions. A lot of the key bacteria that we have problems with that have really high levels of antibiotic resistance, like streptomycin and staphylococcus and E. coli, are actually facultative. And the reason there's so much problem is that we need to be particularly healthy in the aerobic zone for those organisms to not bother us. This actinobacteria group in the facultative, many people in the composting world will know it as actinomycetes, as a common bacteria. This is a facultative one. It does have a general indication when you see that, admittedly, that your oxygen level is probably falling below the ideal. When we start looking at things like fungi, pretty much we've got yeast, which aren't really, they're classified within the fungal family, but they're not actually true fungi. And there's a number of things what we call molds that fit in this category as well. And one of the biggest ones we have problems with are Umicites molds. They're called the water molds. And they thrive in these anaerobic conditions, and they cause things like downy mildew, Peronospora, those sorts of diseases. They occur in this low oxygen environment. All of the fungi that we're really looking for from a beneficial point is that we're actually really only thriving mostly in highly aerobic conditions. Things like protozoa, which are vital to that nutrient cycling, the poop loop we were talking about earlier, they only really work and are happy to survive and live and thrive in these aerobic conditions. And nematodes is a really interesting one, because what the RF means is root-feeding nematodes. So over here, these root-feeding nematodes actually are only about 5% of the nematodes known to man at the moment are root-feeding, therefore pathogenic pest nematodes that feed on the roots of your plants, which actually cause massive economic loss with a whole lot of crops, but they thrive in these anaerobic conditions. So when we drive it into an aerobic state and maintain soil oxygen levels in the soil, and we talked about it last week about how the structure, how you improve soil to get more oxygen in and the benefits of it. But what these are talking about now I've forgotten off the top of my head what the BF and FF. You've got nematodes are, as I said, largely the vast majority of them, 95% of them, are beneficial to our soil system. And most of the nematodes, or most of the root feeding nematodes that cause this problem, are actually controlled by these predator nematodes. They actually consume the vast majority. There are fungi that eat the root feeding nematodes. Even use a little, almost like a little lasso, the nematode will swim through, and the fungi pull tight and then feed on that nematode. So as long as we maintain this highly oxygenated system, this free and friable and open soil, we'll suffer, by for very good reasons, for very few pathogenic or disease issues will cause us problems.
I'm giving you a time. It's now 7:35. There are a couple of questions quickly. The fungi to bacteria ratios, are they biomass weight of fungal to bacteria cells or something else?
That's probably a pretty good question. Off the top of my head, I wouldn't have said it was biomass weight, because fungi are massive organisms in comparison to bacteria. I can't tell you off the top of my head exactly what that measurement is. If you get a soil food web analysis, it will give you those ratios, but I can't tell off the top of my head without looking at that exactly what the unit of measure is. Sorry.
And one more question before we move past this is, and I think it's spelt wrong, it says bacterial feeding and fungal feeding nematodes, but I suspect that means bacteria feeding and fungal feeding nematodes. That's a question,
okay.
Daniel.
Yeah, so there are fungi that feed on nematodes. I can't recall most of the what they call entomopathogenic organisms. So these could be bacteria or fungi, but most of those that feed on other organisms rather than parent material, but they actually feed on insects and other organisms, those are mostly fungi. The few bacterial ones that tend to be entomopathogenic bacteria tend to be feeding more in the soil, in the leaf system above the soil. So the only ones I can tell off the top of my head is, but the main control for nematodes is actually predatory nematodes feeding on root feeding nematodes. But it's a complex system. There's obviously a lot of things happening there in regards to root feeding nematodes, but the key point was essentially if you operate a healthy soil system on this anaerobic stage, we really just don't have a problem with root feeding nematodes.
Thanks, Daniel.
Now, part of this presentation, I'll try and speed up a little bit, is getting people to understand again here we're going to talk about fungal to bacterial ratios. And what we're looking at, what I want you to focus on rather than that whole diagram, because I'm going to flick about 4 or 5 of them past you in a minute, is over here at this end, this represents a higher level of plant growth. And all the way down here as we're looking at a lower level of plant growth. It's measured in milliliters, but essentially what I want you to see is down here is low growth, up here is high growth. And what they're doing is they're wanting to look at various different inputs and how they relate to plant growth. And so in this case, the study was done by Dr. David Johnson, very highly respected soil scientist. He's doing some absolutely amazing work in New Mexico, the university there. So what they did is they tried to look at what are the things that influence plant growth. And there's some very interesting finding here. So this R value essentially is trying to correspond the phosphorus input and how it relates to plant growth. And what they're saying is plant growth, you've got this nice curve runs through here. Really, it's quite a good even curve. What we're looking at in this case is as the phosphorus level goes up and goes down, it has no relationship. The soluble phosphorus levels actually has no relationship to the plant growth, which will shock the fertilizer industry, but that's the truth. So then they did it with potassium. Same thing. This is a potassium level up against the same growth curve once again. The treatment number I'll show later with what they were actually doing down here, but essentially they were putting in potassium, looking for growth, and there was a range of inputs, but there was no relationship between growth and potassium. Obviously, they weren't only putting potassium in, otherwise you wouldn't be getting plant growth. But as they increased the potassium, the potassium level had no relevance. In fact, it was actually one of the lowest points at the highest level of growth. Then they did the same thing with organic matter. It tracks a little bit differently. Why it's bouncing up and down in this particular case, I'm not 100% entirely sure yet, but essentially what you're looking at again is a relationship between your organic matter levels in the soil and plant growth. They did the same thing with nitrogen. What we're looking at is starting with very little low levels of nitrogen, but by the time the plant was growing at its maximum growth rate, what you're looking at is there was very, very little nitrogen. They were measuring NO3, and this is critical to understand. They're only measuring NO3, nitrate nitrogen. It's the nitrogen that causes our ecosystem so many problems when we have an excess. But what they're showing is there's no real direct correlation with nitrate in the root zone with plant growth. So what did create that? What was the one input? What was the relationship that was virtually as linked as you can see on any input, vastly more linked than any other input? Is actually what they're looking here is fungal to bacterial ratio. So this blue and green line is the level of fungi. Up the side here, we're looking at fungal bacterial ratio. And what we're looking at is that as the fungal ratio lifts compared to bacteria, we see a direct correlation basically with healthy fungi in our plant system and plant growth. There's obviously an awful lot going on here, but really what they're trying to do is understand that out of all the various different things that you can control in your soils, that you can put into, that essentially the one thing that is most closely linked to actual plant growth is this all-important fungal to bacterial ratio. Now, quickly on the nitrogen aspect, because this is a very important part of the nitrogen process. What we're talking about is the nitrifying bacteria, which create all this NO3. It functions in soil that has, it goes crazy in soils with relatively high pHs. But when you have fungi present in soil, they tend to drive the pH of the soil down, and so they create a slightly acidic environment. So when you're talking about acidity around 6, fungi are very happy, nitrifying bacteria that do all this conversion of ammonium nitrogens into NO3s, which if we've often seen in our soil analysis lately, we have excess of NO3, and this is partly to do with these nitrifying bacteria. The thing that controls that is fungi. The thing that actually holds the pH in the soil system in a balance and control that stops a whole lot of nitrates flooding the system is actually fungi present in your soil. So even the nitrogen system in your soils is closely managed by that fungi to bacterial ratio again. We won't go into the rest of the nitrogen cycle. It's a separate topic on its own. What we're looking at here is, I quickly flashed this through last week, but essentially what we're looking at is the standard cultivated land analysis of biomass produced per square meter per year, of what they call dry biomass. I would have taken it and effectively dehydrated it and then weighed it, and the average production of cultivated land is only 650 grams per square meter per year, as an example. Across a whole range of crops and cropping systems, but using traditional cultivated land. What we're looking at here is a transitional. So what we've done in the first year is that this is where they start focusing on the microbial network of the soil. They start building and fixing that fungal to bacterial ratio. They stop tilling the soil. They stop doing all those things that do harm and start actually feeding that soil food web and getting that fungal to bacterial ratio right. And this is what happens in year one. By the time that they've been carrying this out for 3 years, this is the sort of biomass production that they can actually achieve or are achieving in a whole range of cropping systems around the world. So you can see, even as I said, tropical rainforests, hugely productive places, massively more productive than what we're currently doing with cultivated land. Our potential is, and it's very important to understand why it's so important that actually we look at, there's an awful lot, we can actually produce an awful lot of food on a much smaller amount of land. And there's a whole range of benefits associated with that.
Dan, you've got a quarter of an hour.
Okay, I'll try and speed it up. So one of the things I wanted to talk about here is essentially looking at, if you want to, most of the soil samples that we've been looking at lately, we've actually got lots of bacteria. The problem what we're looking to do is we need to build up the fungi. So how do you do that? Well, when we talk about feeding, rather than putting fertilizer in and inputs in that are basically feeding and thinking in terms of feeding the plant, what we need to do is switch our thinking into systems thinking, where we're actually feeding the part of the system that we actually want to lift. And in most cases, we're looking at wanting to lift fungal activity in our soil. So things like humic acids, these are complex carbons, complex protein, things like aloe vera, oatmeal and bran are examples. Fish hydrolysate, this is not cooked fish. This is not fish that has gone through a heating process. This is actually using a biodigester, using a biological digesting system. It's not one producing methane. It's one basically using biology to break down the fish. So it's a cold microbial process that gets used. It maintains the fish oil, so it's a bacterial process, but it's the fungi absolutely love that as an input where it's used. So a lot of the time people suggesting put molasses in as food sources, or even using sugar. And what you're really doing here is just feeding the bacteria again. And we need to really be looking at feeding our fungi much, much more.
So in terms of the humic acid, Daniel,
yep.
Now, how can we produce humic acid in our own, farming systems? Okay. And yeah.
Yep. So that brown liquid that comes out the bottom of composting piles, that essentially is humic acid. So if you've got a really good composting system running, and you can actually capture the water that is draining out of that composting system, and there are NZ Box is obviously an excellent system to be able to do that. There are systems there where you can catch it, but that brown liquid that's coming out the bottom of those compost piles, that is humic acid. So that after a while, that can be an extremely useful. If you've got that sitting on the ground, you'll find, as some people have, the soil underneath the compost pile becomes hugely rich, just because that humic acid feeding the biology underneath it. So if you can catch it, sometimes you'll find it, there's quite a bit of humic acid sometimes in worm teas, and in many cases, because you've got a composting process running, there'll be some humic acid in that. But essentially it's what makes the clear water go brown. Once that's the liquid humus, or the soluble humus that's in the system. After a while, of course, if you keep pouring water through it, you'll only get water out the bottom, because the humic acid, you've drained out the soluble component of it.
And you would, again, just to be very practical, that gets watered down, and you would you water that onto your plants or directly onto the soil?
You can do both. Humic acid is beneficial to put over the leaves of the plants. It's feeding the whole biological system. So yeah, it can be used even through into irrigation systems if you're wanting to as well. It's hugely beneficial. Obviously, there's lots of forms of it. Yep.
And going back to the complex proteins again, I'm just wanting people to be able to go away and tomorrow look at what they actually have that they can utilize to start feeding their fungal network. So you mentioned aloe vera. So how would you use that?
Aloe vera is the same as if you take essentially the whole leaf and juice it, I guess is the best description, or crush it. It can be added into water if you can solubilize that and add it into water, and that can just be watered onto the plant and the soil. A very, very useful fungal food. You can also obviously through compost is obviously going to introduce a certain amount of compost coming in. As the first thing it does is it's the humic acids. When you water it in, you'll end up with that humic acid flooding into the soil system below as well.
And we have another great question. Why is solid raw humates, finely ground, not as good as humic acid in liquid form?
Well, you have to use with raw. There's about 15 different humate products that we could talk about in this discussion. Raw humates are extracted usually from brown coal, so potentially, it's useful, but you do have to use a lot of it. It's not soluble at all, so it takes a long time to be broken down, and so the process is somewhat slow in many respects, and you're adding, in that case, it's more about a lot of carbon that you're adding to the soil. Using a liquid humic acid from a compost, or a liquid humic acid, is a much, much quicker way of boosting fungi, and fungi will respond to it in days rather than months. So there are uses for all of these things as far as that goes. The best source of raw humates is from liardonite, which is a different structure to brown coal, but in New Zealand the local sources all come from brown coal. Yes, it has its uses, but it's not as useful in the regards of actually getting a result and waking up a sleepy fungal system.
Yep, great.
Okay, so what I'm going to do is I'm going to run through a quick short video here, because what I want you to understand, this was the fastest way we really get through a very important message, and at the end of all of this is just how important fungi is, not only to our soil and our plant life, but actually to our planet. So we'll see if we can get this to play.
How is carbon stored in the soil? During photosynthesis, plants absorb CO2 and sunlight. They release oxygen and produce sugars, carbohydrates, and other long-chain molecules that are mostly made up of carbon atoms. Plants release up to 40% of these carbon-rich compounds into the soil as exudates to feed fungi and bacteria. They do so in order to promote growth in the populations of these beneficial organisms, as this will eventually result in a return of nutrients to the plant. Please watch the animation on nutrient cycling for further information. Fungi produce mushrooms on occasion, but that's just a small part of the way they grow. The majority of the fungus consists of hyphae, long, narrow tube-like structures that are made mostly of carbon. As a fungal hypha grows, it gets longer and branches off in different directions, looking for new food sources. Over time, its walls grow thicker and thicker, storing more and more carbon as the fungus continues to absorb exudates from the plant, and also organic matter in the soil. So how can this help in the battle against climate change? Well, the biggest single organism ever discovered on planet Earth was a fungus in Oregon that measured approximately the size of 1,665 football fields and is somewhere between 2,000 and 8,000 years old. That's a lot of carbon being stored up for long periods of time. So you can think of this as a kind of carbon pump. Atmospheric carbon is absorbed by plants and pumped into the soil, where it is stored for hundreds of years by fungal hyphae. This process is known as soil carbon sequestration. How effective could soil carbon sequestration be in the struggle against climate change? Well, estimates vary, but here are some reasonably conservative numbers. Atmospheric carbon levels are currently just over 410 parts per million. That's totally off the charts when you look back over the last 450,000 years, as climate scientists have been able to do using ice core samples. This number has not been above 300 parts per million in that time. The red line on the graph is temperature change. As you can see, there has been a clear correlation between temperature change and atmospheric CO2 over the last 450,000 years. If the temperature continues to rise in accordance with current CO2 levels, then we're going to see some dramatic changes to our habitat. Just to put that in perspective, humans have only been on the planet for around 200,000 years, so we have never seen conditions like those currently being modeled by climate scientists. What is considered a safe level of atmospheric carbon? According to the United Nations IPCC, this is somewhere around 350 parts per million. That's around 60 ppm that need to be removed from the atmosphere, which equates to approximately 450 billion tons of CO2 equivalents. We're going to need this number in just a minute, so let's put it over in the corner here. How much of this can be sequestered into the soil? The answer is, we don't know. What we do know, however, is that it is possible to sequester in excess of 10 tons per hectare per year, and possibly as much as 20 tons per hectare per year, as demonstrated by Dr. David Johnson at New Mexico State University, who has been successfully restoring the soil food web. Yes, this is cutting-edge research, but with more investment, this could possibly become achievable worldwide quite quickly. Humans manage approximately 5 billion hectares worldwide. So based on 20 tons per hectare per year, that equates to 100 billion tons per year. Total greenhouse gas emissions for the entire planet in 2019 were around 37 billion tons. About 50% of emissions get absorbed by natural processes, according to research by NOAA, leaving around 20 billion tons in the atmosphere. So if we did nothing else but regenerated the world's soils, we could potentially sequester about 80 billion tons per year. 450 billion tons divided by 80 billion tons per year means that we could theoretically get back to 350 parts per million, the safe level, within 6 years, just by regenerating the world's soils. Of course, we cannot do that overnight. Allowing time for global implementation, 10 to 15 years is a more realistic time frame. If you factor in reductions in greenhouse gas emissions, the picture begins to look far more optimistic. The bottom line is that this could be a big piece of the puzzle when it comes to fighting climate change. For more information about the numerous benefits of the soil food web and how you can get involved, visit soilfoodweb.com.
Okay. I'm just trying to get my mouse to operate.
So, and that's from Elaine Ingham's Food Soil Web, and she did her original study from the Rosedale Research Institute. So there's a great legacy of these scientists who have been, collecting data around these processes for some time, that we're really grateful to be able to build on their knowledge and share it with you today. There was a question just before we go, because we're about to round this up. We are almost on time, but we did start at 10 minutes past, so we will go a little longer for your questions. But if you were going to use the aloe vera, what would you dilute it to? Is it about 1 to 10? What would you recommend, Daniel?
It's be where I would start, 1 to 10, but you could probably dilute it much more than that.
And it's also a very good pest. It stops animals eating the plants, for example.
Yeah, if you spray aloe vera onto plants for kind of reasons that we don't entirely understand, that it actually stops grazing animals often feeding on the plants, or birds causing plant damage. They use it in Australia against kangaroos and rabbits, spraying aloe vera. But aloe vera itself is known as one of the greatest fungal foods on the planet. So it's a bit hard to understand why animals don't like eating it, but apparently they don't.
So does that take you to the end, Daniel? Is there anything else that you want to share with us tonight before I wrap up for us?
No, I think we can wrap that up, and unless we've got other questions to deal with, but otherwise we'll wrap it up.
So I just really want to thank you guys for joining us on this, in our masterclass series. I really encourage those of you who haven't yet seen The Farmer in the Field, where you get to take this kind of abstract kind of knowledge and see how it's actually being utilized by farmers as they're transitioning their farms across. So the three key takeaways are the ability of soil fungi to create real, it is the ability of soil fungi to create real ecosystem transformation. There's a realization that it's not about us going back to what our grandparents did, but that the real innovations around regenerative practices are about creating highly productive and profitable no-till farms. And the thing that we need to be focusing on is actually our bacteria to fungi ratios. But these are the keys to our farms being successful without harming nature. We are going to be offering our third masterclass series, which is going to be looking at plant health, looking at the health of the plant from an above ground perspective, looking at managing symptoms, disease symptoms, and increasing photosynthesis while we're recovering our soil and also saving the planet. I really want to thank you very much for being somebody who is interested in learning these things and actually applying them in your projects. Together, we are really able to make a difference, and we are looking for systems change in 3 years. So thank you very much for joining us. If you have any questions, just chuck them up. But otherwise, we will wrap this up. You can go and find this in our YouTube channel. Please sign up for the YouTube channel so that we can get our own site. It's easier for us to then share it. And please let people know in your lives that this resource is available. If you're somebody who has capacity to fund the work that we're doing, we would be really appreciative. We have done this because we know that it is needed, and we're out there in the world currently looking for the funding for us to continue. Thank you very much.